York City rats may be pizza-nabbing pests to many, but they're paving the way in our understanding of evolutionary biology in our changing world. The world is becoming increasingly urbanized, and forests are being cut down and replaced with cities, asphalt, cars, and people. And as someone who loves nature and biodiversity, it is disheartening to see. But New York City rats show that nature, that evolution, doesn't stop when we get somewhere and mess everything up. We may present rats with new pressures, but they adapt and change to fit the ever-changing urban landscape they also call home. And if you ask any New Yorker, these little guys are thriving, maybe too much. That is what makes New York City rats, or brown rats, Rattus norvegicus, the poster child of adaptation to urban environments. Cue Harbeck et al. With their study on genetic adaptation in New York City rats to answer the question, did genetic adaptation play a role in the spread of rats in cities? Scientists have already studied how urbanization changes gene flow and therefore increases the effect of genetic drift. They have also begun to identify phenotypic differences between city animals and their country counterparts. There have even been studies that have identified mutations in urban rats that have been experimentally shown to confer anticoagulant resistance, which is the mode of action of popular rodenticides, including warfarin. Where Harbeck et al. Come in is in searching for evidence in the genome of positive selection for these adaptations for city life, because, intuitively, adaptations like resistance to rodenticides seem like they would be positively selected for, but our intuition is not proof that positive selection is occurring. Instead, Harbeck et al. Compared the whole genomes of 29 New York City rats captured from all over Manhattan with the genomes of 9 ancestral Chinese rats captured from Harbin, Heilongjiang Province, China, to look for selective sweeps, which would be regions with high haplotype homogeneity among the sequences that differed between the New York City and Chinese rats. Panel A of Figure 2 shows how Harbeck et al. Found their genes of interest through whole genome analysis. First, they determined selective sweeps through G12 and H-scan measurements, both of which measure homogeneity of haplotypes in the sample around focal SNPs. This figure shows 3 loci that experienced various levels of sweeps. In panel A, you can see a random locus. There's no consistency, you can look up and down and it looks completely scattered, so this is no evidence of a sweep. In panel B, you can see evidence of a hard sweep, so if you look up and down, you can see that there is a lot of consistency for most of the samples. In panel C, you can see a soft sweep, so they show some consistency, not most, but definitely a sign that a sweep was present. And if you look in panel D, you can see that is panel C compared with the Chinese samples, so the Chinese samples are highlighted in yellow, the New York samples are highlighted in purple, and if you look comparatively, there's a lot more homogeneity in the New York samples rather than the Chinese samples, indicating that a sweep was present in the New York samples that was not present in the Chinese samples. After G12 and H-scan measurements were used to find areas of haplotype homogeneity, FST measurements were used to determine genetic differentiation between the two populations. The results of this search are shown with respect to the G12, H-scan, and FST results in purple, blue, and pink respectively. The compilation of the 3 factors was used to find the most probable areas of positive selection in New York rats. To find potential phenotypic effects of the mutations they found, they looked for protein coding sequences less than 20 kb away from the SNPs they identified from the G12, H-scan, and FST measurements. These known protein coding genes are labeled in the chart with larger circles and a label with the gene's identity. The protein coding genes they identified were primarily associated with diet, nervous system, metabolism, and locomotion. The hypothesized pressures that selected for those adaptations are summarized in panel C of Figure 2. The increased fat and sugar in human trash, which is New York City rats' primary source of nutrition, might select for mutations in ST6, GAL, NAC1, CHST11, and GBTG1, which are linked with oligosaccharide or carbohydrate metabolic processes. The urban landscape and risk from pedestrians and vehicles may have selected for mutations in FGF12, EPHA3, and CHAT genes, which are involved in locomotion. Another locus of interest they identified was in the gene CACNA1C, which has been implicated in genetic studies of psychiatric disorders such as anxiety in humans. So, if it makes you feel any better, the stress of city life might be getting to rats too. Interestingly, the VKORC1 gene, which was previously identified as the mutation that confers some degree of anticoagulant rodenticide resistance, was not among the loci identified in this study. Although they did find substantial differences between the New York City and Chinese rats at this locus, they hypothesized that the lack of fixation of the VKRC1 mutation was due to the phasing out of warfarin as a rodenticide in the 1970s. And VKORC1 is not as protective against second-generation anticoagulant rodenticides, or SGARs. Maltsev et al. Similarly found the VKORC1 gene mutation in low percentage among Moscow rats. Unlike Harbeck et al. With regard to the New York City rats, Maltsev et al. Attribute the lack of fixation in the Moscow rats to the minimal use of anticoagulants. In both the New York City and Moscow rats, the lack of fixation of this mutation may be attributable to the weakness of rodenticide as a selective pressure. Despite being a genome-wide study, there are still other differences between urban and rural rats that weren't identified in this study. Puckett et al. Found altered cranial shape, longer noses, and shorter upper molar tooth rows in city rats as opposed to the ancestral Chinese rats. Puckett et al. Hypothesized that the longer noses are in response to the colder New York climate and the shorter tooth rows are in response to the high quality and soft diets, pizza and donuts over nuts and seeds. Although not in rats, but still in rodents, Maza et al. Looked at differences in behavior in urban voles opposed to rural voles, and found that urban voles had greater behavior flexibility, allowing them to exhibit more exploratory and bold behavior in situ. Neither the altered cranial shape nor any behavioral differences that were identified by Puckett et al. And Maza et al. Respectively were identified by Harbeck et al. This highlights the weakness of Harbeck et al.'s study, which they readily acknowledge. An issue with looking at SNPs is this technique is relatively blind to complex polygenic traits like cranium shape and behavioral flexibility. And unlike Maza et al., Harbeck et al. Did not sample rural New York rats and only used ancestral Chinese rats for comparison. This is a weakness in the study, as selective sweeps cannot be directly linked to urbanization instead of the New York environment as a whole. Additionally, they identified the target protein coding genes by picking genes that were within 20 kb of the mutations they found in their analysis, but they didn't prove that those mutations actually affect the expression of the proximate protein coding genes. However, despite these weaknesses, it was still a strong study and did identify several loci that could potentially be the focus of future studies. Additionally, they found genetic evidence of selective sweeps occurring in New York City rats, and highlighted the genetic difference between the New York City rats and the ancestral Chinese rats. And the implications of the study are incredibly significant. Many of the genes implicated, especially those that affect psychiatric health and diet, are relevant to humans who are also adjusting to urbanized life, many of whom suffer adverse side effects as a result. Additionally, as our planet becomes more urbanized, it is important to know and understand the ways in which animals adapt and change to survive in this new world, and how we can maintain biodiversity in spite of humanity's destruction of wildlife and their habitats. At the end of the day, there is something that is a little comforting about knowing that nature persists in spite of everything we've done. In the words of Dr. Ian Malcolm,
life finds a way.
Thank you for watching, and thank you to all the amazing researchers who have contributed to our understanding of biology, evolution, and the effects of urbanization on the animals around us.